In this portion, we're going to talk about the regulation of urine concentration and the volume of urine. So how much urine will be excreted per day. This is regulated by several hormones, for example, aldosterone and cardiac natriuretic peptide and antidiuretic hormone. So let's take aldosterone, for example, and one of the main stimuli for aldosterone secretion would be hyperkalemia, which is the increase in potassium levels in blood. What the function of aldosterone, and I'll mention it in scientific terms and then explain it. So the function or the effect of aldosterone on the kidney is that it increases the tubular reabsorption of sodium and increases the tubular secretion of potassium, which means that increasing the tubular reabsorption of sodium, that means we're going to keep sodium in the body. It'll prevent the loss of sodium in urine. And when you keep sodium, water is going to follow by osmosis. Okay, so an increase in levels of aldosterone is eventually going to lead to increase in blood pressure. The other function that it did was the increase in tubular secretion of potassium, which means we're going to be dumping more and more potassium into urine. And that, remember, the main stimulus for the release of aldosterone was hyperkalemia, and aldosterone is going to increase the dumping or the loss of potassium in urine, bringing that potassium levels down back to normal. And the way I remember it is that it keeps the sodium in the body and dumps the P in the P being potassium in the urine. As for cardiac natriuretic peptide, that's a peptide hormone released by the atrium in response to high blood pressure. And from the name, you could guess the function of it. It is going to dump sodium in urine. And again, when you dump sodium in urine, following it would be water by osmosis, and that leads to a decrease in blood pressure. Now, the function of ADH, antidiuretic hormone, if you guys remember that hormone, it was made in the hypothalamus and released from the posterior pituitary. The ADH is going to be released in response to high blood osmolality, meaning that there is just too much salt in our blood. Okay, so let us just get a background on the effect of ADH on the distal tubules and the collecting ducts. Under normal circumstances, when there is no ADH, the distal tubules and collecting ducts are not permeable to water. Okay, so any urine that is going to be made will be pretty much diluted urine. But in the presence of ADH, the distal tubule and collecting segments are now going to be permeable to water. So water would be reabsorbed by osmosis, will keep the water in the body, and that way we are going to be concentrating the urine. So we'll take a look at the effect of ADH on the urine volume. So again, normal circumstances, we've got low levels of ADH, and you can see here, distal tubule and collecting ducts are not permeable to water. We are going to make our normal kind of diluted urine. Let's say we ate a bag of salty chips. Now our blood osmolality levels are high. That will release ADH from the posterior pituitary, increase the permeability of the distal tubule and the collecting ducts. And you can see here that now water is being reabsorbed into the interstitial fluid, and then eventually will get into the capillaries and into the bloodstream. And that is why increased salt intake will lead to an increase in blood pressure, because we are now concentrating the urine in order to keep that water in our bodies and balance all of that salt that we just ate. Okay, so takeaway from this, normally low ADH levels, we make diluted urine because the distal tubule and collecting ducts are not permeable to water. Under high levels of ADH, the distal tubules and collecting ducts become permeable to water, and water is reabsorbed through osmosis because of the hypertonic medullary interstitial fluid. So I want to take a moment here and talk about that. First off, what does it mean? So the interstitial fluid in the medulla, remember the kidney was made of the cortex and the medulla, so this is the medulla we're talking about, is normally hypertonic. Okay, so that hypertonicity of the medulla, the interstitial fluid in the medulla, helps with the process here of reabsorbing or pulling water out of the distal tubules and the collecting ducts in the presence of ADH. The hypertonic interstitial fluid is due to the presence of urea in the medullary interstitial fluid. That's how that area is always hypertonic. And that takes us to the countercurrent multiplier. Okay, that's why I kind of wanted to go over that one point, because it is the hypertonicity of the interstitial fluid in the medullary area that is going to help us understand how this countercurrent multiplier works. Two other things that we want to talk about, kind of like background information that we would need to know, that the countercurrent multiplier occurs in the nephron loop. The descending limb is only permeable to water, but it does not allow for the passage of salts, while the ascending limb is impermeable to water, meaning it does not allow the passage of water, but it does allow the passage of salts like sodium and chloride. We also know that the interstitial fluid in this area is hypertonic. So let's take the passage of the filtrate, and I don't want to focus on anything in the filtrate except for the sodium, chloride, and water. Now, what occurs in the proximal tubule, we mentioned that there is the active transport of sodium and water, so that at the end of the proximal tubule, the filtrate there is isotonic. Now in the descending limb, as that isotonic fluid goes down the descending limb, that is only permeable to water. So water is going to leave the filtrate. That means we are concentrating the filtrate. So at the end of the descending limb, that filtrate now is hypertonic fluid. That hypertonic fluid enters into the ascending limb, which only allows for the passage of salts like sodium and chloride ions, but it is not permeable to water. So at the very end of the ascending limb, that fluid now is hypotonic fluid. That hypotonic fluid enters into the distal and collecting ducts, which again, under normal circumstances, we have low ADH levels, so we are going to make diluted urine. Only if we need to concentrate it, that's when our posterior pituitary will release ADH levels, opening up the pores, the aqua pores of the distal tubule and the collecting duct, allowing for the reabsorption of water, leading to the elimination of concentrated urine. Okay, the countercurrent multiplier again is just the portion in the nephron loop, the descending and ascending limbs. And because the direction of flow of fluid is in the opposite direction, and you start off again with isotonic, you end up with hypertonic at the end of the descending, and then at the end of the ascending, you end up with hypotonic fluid. So all of that exchange, or the movement of fluids in the opposite direction, with water being reabsorbed on one end and salts being reabsorbed on the other end, that is where the name comes from, the countercurrent multiplier. So extremely important to remember: descending limb permeable to water only, ascending limb permeable to ions only, mainly sodium and chloride. Again, at the end of the proximal tubule, we end up with isotonic fluid. That's what enters into the descending limb. Reabsorption of water in this area, ending up having the filtrate becomes hypertonic, and then the hypertonic fluid enters into the ascending limb. That is only permeable to sodium and chloride ions. meaning that we are removing salt but not water, so we end up with hypotonic fluid at the end of the ascending limb. Now, a little bit about urea and uric acid. Urea is produced by the body as a result of the breakdown in the catabolism of protein. Urea, we've learned, is extremely important in maintaining the hypertonicity of the interstitial fluid in the medulla. Uric acid is a byproduct of the metabolism of nucleic acids. These are normal products that occur in the body, but we do not need them in the body. So both of these products are filtered through the glomerulus. They both undergo some sort of reabsorption followed by tubular secretion. And at the end of the day, 20% of urea is excreted in urine, while about 10% of uric acid is excreted. Now for the composition of urine. Okay, so about 95% of urine is water, and along with water we have some metabolic waste products like urea, uric acid, and creatinine. Creatinine is a normal byproduct due to muscle breakdown, so there is a normal wear and tear of muscle cells, and during the breakdown of the older muscle cells, we make creatinine, which will be excreted in urine. And then sometimes we have little traces of amino acids, some electrolytes in there, things that we should not find in urine. For example, blood cells, so RBCs, platelets, white blood cells. If we do have RBCs, that is known as hematuria. That should not be there. If we have white blood cells, that means that there is some kind of infection in the urinary system. Platelets should not be found in urine either. Glucose is something that is an abnormal finding. So the presence of glucosuria or glucose in urine indicates that this patient is diabetic and they are not controlled. Obviously, the amount of these products, especially water, water and salt, really depends on the needs of the body. It depends on the amount of water intake throughout the day, even the amount of the kinds of food that we're eating throughout the day. So, we see how urea, for example, was the byproduct of protein catabolism, while uric acid was a byproduct of nucleic acid metabolism. So again, depending on what we eat, if we eat high levels of protein in the diet, you'll find more urea being excreted, for example. The normal amount of urine per day is about a half a liter to about 2.5 liters per day. Again, very variable depending on the amount of fluid intake, the physical activity, and other environmental factors. Renal clearance is the rate by which a chemical is completely removed from plasma by the kidney. Meaning that, for example, if somebody is injected with a substance, how long is it going to take the kidney to eliminate the body, to get rid of all of that substance through urine? It is a reflection of kidney health, so it indicates kidney efficiency. It also helps in the diagnosis of glomerular damage. And we can also use it to figure out the progress of renal diseases. So is the kidney getting better or is it getting worse? And there are different renal clearance tests that are used. There's an inulin clearance test. Inulin is not a normal product that we have in the body. It has to be given to the patient. It is a polysaccharide that is found in the roots of some plants. Creatinine clearance test, though, is the most common test to be done because, again, creatinine is normally produced in the body. Both of these substances, whether it's inulin or creatinine, both of them only undergo glomerular filtration. And that's why we could use them to figure out glomerular health and glomerular damage, because they are only filtered in the glomerulus. There is no tubular secretion or tubular reabsorption of inulin and creatinine. And this ends this part.